We like to study antibodies in the context of viral pathogenesis. And I think that's what makes our lab a little bit unique, is rather than saying they protect or they don't protect, we like to try and interrogate how they actually are providing protection by using what we know about the virus in terms of the immune-mediated pathogenesis or the types of tissues that it tends to go to really understand what leads to a protective response, and then can we make it better?
Hello, everyone, and welcome back to People, Parasites, and Plagues, a podcast aimed at delivering information about the fascinating pathogens among us from the scientists who study them. I am Kim Klonowski.
And I am David Peterson, your host for today's episode. Do alphaviruses really think they are the coolest viruses? Today's guest knows the answer to that question and more.
Our guest today is Dr. Julie Fox, Chief of the Emerging Virus Immunity Unit, Laboratory of Viral Diseases at NIAID.
And I have to say, first off, that that's a whole lot of acronyms because you're the chief of the EIVU within the LVD at NIAID at NIH.
Yeah, you also forgot I'm within the DIR. Yeah.
Okay.
So throw one more in there.
What is the DIR for anyone who's interested?
The Division of Intramural Research.
Oh, okay. Gotcha.
We will remember that. Tell us about your pathway to science. And because we love asking this question because we get so many different responses and they're all really fascinating. So when did you decide that a career in science was it?
So this goes all the way back to undergrad. I always knew I liked being in biology, but I thought really the only career path would be into medicine and medicine or like pharmacology. And part of the curriculum was to take a microbiology course. And I loved it. I loved streaking plates. I could streak plates all day long. I could make cultures. I enjoyed it so much. And it wasn't until I was working at a restaurant, I was a server, and there was this older guy who was also a server, and I was chatting with him about, going into medicine, but how much I loved microbiology. And he goes, you could do that as a career, right? And I was like, what are you talking about? He's like, the people that are teaching you microbiology, they probably have labs and you can go work in these labs or you could have your own lab. And it never clicked to me that could actually be a profession that I could do. And from then I started researching. I started reaching out to professors asking if I could volunteer in their lab or to work in their lab. And I eventually started I got an internship at a water quality company with the utilities. And I worked in their microbiology lab. I worked in their research part of the lab, and I just absolutely loved it. And at that point, I was set on getting a PhD and trying to run my own lab.
And where did you do your undergraduate?
At the University of Central Florida.
Okay. What an amazing random encounter that set you on your path.
I mean, for all I know, if I didn't have this one particular conversation, I probably would have thought I had to go and become an MD to stay in science.
Well, thank goodness for that conversation.
Exactly. The least likely places, right? Sometimes you just never know. Okay, Julie, so you finished your undergraduate degree, and then you decided to do the PhD, which landed you here. So you started working on viruses at that time. So not necessarily streaking plates, maybe doing some plaque assays. So you had a switch then. So how did you decide to go into the lab that you did here for your PhD work?
So even back in undergrad, I was really interested in viruses. I took a virology course. It's just that a lot of the labs that were there wasn't a lot of virology labs that were taking students at the time. And so when I applied to UGA because I wanted to get a PhD in infectious diseases. And the labs I chose to rotate in I picked two virology labs because deep down inside, I felt viruses was my path forward. But I also picked a parasitology lab because when I worked at the water quality plant, I worked on Giardia. And I liked working on parasites, so I figured that was almost a backup just in case I didn't like viruses. And so during all the different rotations, I really discovered I enjoyed viral immunity a lot and vaccinology and talking with people. My previous mentor at the water quality plant, I gave him all these options. I was talking with him about it. And he's like, vaccines are something that are always going to be necessary. Having that background and being in that field is going to be something that is going to be beneficial in the future, having that experience. And so that's how I ended up in Ralph Tripp's lab working on influenza virus and studying viral immunity in that context.
So after that, you did a postdoc, which is usually required for like the next step into academia or government, not necessarily business. And after that, how did you go from there to NIH? Were you just looking at the want ads and found a position?
So they had posted a position for, they were looking to hire a new tenure tracker, a tenured investigator that studied emerging viruses using animal models. And it like described what I worked on to like a T. And so I applied to an open ad for this position at this, within the LVD.
So how was that transition?
So it was interesting because it happened during COVID.
Yeah, I did note that on your CV. That year stands out like a sore thumb.
Yes, this is really funny. So I accepted the position, I think in December of 2019, and we were getting everything all set up. And then I had planned to go out to do like an initial site visit to kind of plan things. And it was set for early March of 2020. And a week before I was supposed to leave, they canceled the flight. And then I never got to visit until I just moved there and just started my lab in October of 2020. Luckily, there were people there, like our lab chief at the time. He was very helpful in helping me stock the lab. His people were there he would kind of just tell me generally, like, where do you want to put the centrifuge? And so I would do this all remotely. That way, when I arrived, there was actually things in the lab and I didn't just come to a blank space. But yeah, it was, I think the hardest part was just transitioning from a postdoc to a faculty position. It's something that you don't really get a lot of training in, and you have to learn very quickly. Otherwise, you're not going to be successful in that case. And I think it became extra hard because it was COVID. No one was in the building. Besides the trainees who were there doing their work that they were allowed to come in to do. And so I had to kind of navigate a lot of the area or figure out who I was supposed to be talking with, all virtually.
It is kind of sink or swim when you start. I think it's the same in academia.
Yeah, I feel like you always are training for something, and then you move to the next step and you're doing a different kind of training, right? And you're always still training, right? And you take that jump to the PI position, and I feel like there's just a lot. Unless you had a postdoc PI that maybe let you, for example, write some animal protocols, do some things that kind of give you a little bit of a head start.
Well, I think part of that is wherever you end up moving to, like for me, going to the LVD, they were so supportive and helpful. And I think that really made a huge difference, especially with our lab chief. If like you said, an animal protocol or like, oh, I know I have to register my pathogens. How do I do that? Mm-hmm. And so I could go to him and he would really be helpful in telling me, okay, we need to contact this person. We need that person. And I think if it wasn't for him, I probably would have struggled a lot more in the process.
And I should mention that one of the reasons we invited you here, of course, the chief reason is your cool science. But second, you spend a lot of time talking to our graduate students and I guess mentoring them about the transition from graduate school to maybe postdoc and on to NIH. So thank you for doing that.
Of course, it was actually really fantastic. They were a lively bunch. They had a lot to say, whether it was questions about their own path and how to get there, or just telling me about their project. There's really exciting work happening here. I'm going to have to go and search some of these papers up on PubMed when I get home.
Cool.
So if one is interested in a career in government research, what advice did you give maybe the students or you would give someone else?
So the NIH is unique in that it's very academic there. We do have, obviously, it's still a government position, so there are the other additional like red tape that you would hear about. But generally speaking, it's a fantastic place to do science, and we don't really have we have the flexibility to be able to do kind of similar science that would happen in a university. But in terms of getting into it, I don't think there's really anything more or anything different than applying to a university position. The only difference is that I don't really do a lot of teaching. So I didn't have to worry about writing a teaching statement or trying to propose a course. It was all mostly based on my research and what my plan was in the future.
And some people do go into the NIH as a postdoc too. It's not just as a faculty position like yours.
Oh yeah, we have the postdocs are one of our main workforce at the NIH, and they're fantastic. And I recommend people go to the OITE website. You can look for open postdoc positions. We also have a large post-bac community there. So these are people who finish their undergrad and they spend 1 or 2 years, sometimes 3 years, doing full-time research in the lab and preparing themselves to go to graduate school or going to medical school or some professional school.
I think we have at least 2 graduate students in our department who came from the NIH post-bac. And I have to say, they came really prepared, really well-trained. So,
well, yeah, I mean, they come in, they're almost like a first and a second year graduate student at that point, but they don't have the classes to distract them. So they're in the lab full-time. If they're in a great lab, they understand the process. They know why they're doing it. They have the time to be able to apply this knowledge and get a deeper understanding of why they're doing things rather than just learning the skill, which I think is really important to succeed well in graduate school.
Yeah, absolutely. And I didn't realize these were multi-year long programs. For some reason, I only thought it was like a summer program.
We do have the summer internship program. That is mostly for current undergrads. They can come in for the summer and do research. But the post-bac program, it's at least a 1-year commitment. But most people like to do it for 2 years just because by the time you start, you want to apply for positions, you haven't really been in the lab very long. And so what kind of experience have you gained out of that? So doing the 2 years, you get 1 full year before you start applying for graduate or professional school.
Well, I think with that, we can move on to alphaviruses. So I guess we should address what I mentioned in the lead-in. Are they the coolest viruses?
I think they're the coolest viruses for, especially to study. They are really easy to work with. And I think that's one thing that models always benefit from. They can grow to really high titers in cell culture, so you never have to worry about, am I going to have enough virus to do this experiment? They have pretty decent mouse models. They're not perfect, but what animal model is perfect? And they're easy to manipulate. Like we have, you can introduce like GFP or anything into their genome, and usually they can take it in. So, and there's great tools. They've been around. People have been studying these for decades. So I think that's why we have a lot of great resources to study them.
So let's just talk about alphaviruses in general for people who are not familiar with them. Do you want to throw out the names of some alphaviruses that maybe some scientists out there might be able to latch on to and be like, I know that virus?
Yeah. So, I mean, I pretty much put out there that alphaviruses are mosquito-transmitted viruses. And the one that we study a lot and is probably the most well-known is chikungunya virus. It does not infect chickens, so people think that. But it causes severe polyarthritis and polyarthralgia. And this is during acute disease where you first get infected, your joints can swell up. It's really incapacitating. And then for a subset of people, in some cases, it can be between about 25% for the recent outbreak in Central and South America. They can develop this chronic disease where they have this persistent joint pain that can last for years following infection. And so it really can have a dramatic impact on the economy as well as people's social economic.
That's a really unique course of disease that it attacks the joints. You think of viruses, I guess if you ask somebody what viruses do, they'd probably say, well, they affect the lungs because a lot of viruses are, transferred by coughing or something. But the fact that they target joints.
Well, I think if you think about it from the immunology point of view, is the virus actually targeting the joints, or is it the immune complexes or something else that's causing the disease?
So the virus is definitely joint tropic.
Okay.
I don't know why it likes going there. That's a fantastic question. We should look into it. Maybe it's like receptor usage or more privileged site. The big thing is obviously, like you were saying, coughing is a great way to transmit it. But these viruses also cause high viremia, so they just disseminate throughout the body. And chikungunya virus in particular, humans can be, they're an amplifying source. So they get high enough viremia in their blood that a mosquito can get reinfected or infected from biting an infected human.
Oh, that's interesting because usually it doesn't go.
Yeah,
well, I mean, I guess with malaria, it goes into the mosquito, but not for most viruses, right?
Yeah, a lot of the other arboviruses, such as like West Nile virus and even some of like the encephalitic alphaviruses, humans are a dead-end host. But for chikungunya, the level of viremia is so high that we can also maintain an outbreak in like an urban setting.
So in addition to chikungunya, there's the equine encephalitis viruses, and there's how many of those, 2 or 3? I know in the U.S. we have Western and Eastern, but are there other ones as well?
Yeah, so there are other, there's different serocomplexes, and they fall within the Eastern, Western, or Venezuelan. We have Eastern and Western in the US, although we haven't really had a lot of cases of Western in, I think it was like 15 years, although there has been a recent outbreak down in South America. But we do have viruses that are related to Venezuelan that are here. They don't really cause a lot of disease in humans. So it is possible there would be an emerging virus that would be related to one that's primarily found in South America that's already here.
So with that, I think I should introduce a little tale. So when I was a graduate student, I came down with something that gave me a fever. I had this headache that was incapacitating, and my wife took me to the health center at the UC Irvine where we were, and they kind of just sent me home and said, yeah, you have one of the equine encephalitis viruses, which to my mind means ICU, but now they just sent me on and said, yeah, just lie in bed, which is really all I could do at that point, in a dark room, not moving. I don't know which one it was. I don't think they tested. I don't know if there were tests then. What do you think I had?
Remember, she did not take the medicine pathway.
Yeah, thank you for that. I mean, if you were out in California, it probably would be Western equine encephalitis virus, unless you recently had traveled to the Northeast where Eastern equine encephalitis virus is located. It's also possible it could have been like West Nile.
Well, this would have been in the early 80s.
Oh, so pre-introduction. Yes. Okay.
I also read that there's a chance of neurological sequelae, which now has me really concerned.
Concerned or explains a lot. Sorry.
Both. These are two separate viruses, and I guess we should also get into immunity here. If you get infected with Western equine encephalitis virus, does that protect against, well, I guess first reinfection, or protect against infection with Eastern equine encephalitis virus?
So that's a great question. You should be protected against Western. You should not be able to get reinfected. Usually infection with these alphaviruses induces a really potent IgG or antibody response. And because they are systemic, they would become in direct contact with these antibodies upon reinfection and most likely be able to prevent at least any kind of serious illness afterwards.
But no cross protection?
So you could potentially see cross protection. I don't know of any study where they evaluated immune serum from like a Western infected individual. For the arthrodogenic alphaviruses, such as like chikungunya and some of the other ones that are around there, such as Mayara, which is in South America, or like Ross River, which is in Australia, immune serum is cross-reactive to these other alphaviruses, although it's not universal. Sometimes an infection with one particular virus the antibodies will bind and protect against a different alphavirus, so maybe it's not the other way around. So I don't know in that case from Western to Eastern. It's quite possible that it would provide at least some kind of protection. If it was complete, I don't know.
Well, at least that's good to know.
So, Julie, what's the major question of your research?
So we like to study the role of antibodies in protecting against alphavirus infections with the hope of eventually exploring other viral models. And we like to study antibodies in the context of viral pathogenesis. And I think that's what makes our lab a little bit unique, is rather than saying they protect or they don't protect, we like to try and interrogate how they actually are providing protection by using what we know about the virus in terms of the immune-mediated pathogenesis or the types of tissues that it tends to go to really understand what leads to a protective response. And then can we make it better? Can we try to enhance where like an antibody is going? Can we induce some activity like Fc effector functions? Is that going to enhance the protective nature of the antibody? And if we understand how it is already working in the context of the pathogenesis of the virus, it might help us kind of narrow down what the next step would be to find these optimal therapeutics.
I had a really simple model in my head about how antibodies work against viruses. But that was, and maybe it comes mostly from influenza because you measure whether antibodies are neutralizing, meaning to my level of understanding that the antibody binds the virus and prevents it from infecting. But you were talking about a number of other mechanisms by which antibodies can affect the infection. So how do those other ones work?
So in the case for alphaviruses, like you mentioned, neutralization, which is, like I always say, is what everyone always thinks about an antibody can do. But there's a lot more literature out there that non-neutralizing antibodies can be just as protective. And so these are antibodies that would still recognize the virus, but it wouldn't prevent it from infecting a cell. And so I mentioned in the case for alphaviruses, when a cell is infected, the proteins that the antibodies recognize would be on the surface of the infected cell. And so we are able to then have these antibodies bind to infected cells. And this kind of protects in multiple ways. You could have, since the virus buds off the surface of the cell, these antibodies can prevent this virus release. So even though the cell gets infected, you don't actually get progeny virus come out, or you get reduced progeny virus. The other thing is you can get, in the context of a whole immune response, there's a lot of immune cells that can engage with these antibodies that are bound to infected cells or bound to the virus themselves. And you can enhance clearance. This is be through engagement of Fc gamma receptors. You also can get additional clearance of either the particle or the cells by complement. You also can get enhanced neutralization with the presence of complement. And so there are ways that the body, in addition to making these antibodies, can now almost amplify the protective nature by doing these alternative approaches to either clear infected cells or to alter the immune response that's coming in.
In terms of the Fc response, okay, so a pathogen in some ways would not want a neutralizing antibody response. If the pathogen had a brain, it would say, I don't want to have to go against a neutralizing antibody response. Or am I wrong?
So the one thing that is, I mean, obviously a neutralizing response, if it's potent enough and you prevent the infection, that's fantastic. And obviously a pathogen, if it had a brain, it wouldn't want that, right? It's the most easy way to stop the infection. But in case of alphaviruses and a lot of viral infections, you can get rapid generation of, they end up putting in a lot of mutations within their genome just because of the error-prone nature of their RNA-dependent RNA polymerase. And if you have a neutralizing antibody, you potentially could start generating escape variants. And we saw this with the COVID and the Regeneron antibodies.
Oh, yeah.
I forgot what they're called, but the names of them.
So this was the monoclonal treatment?
The monoclonals, yeah.
Okay.
That they would give to people. And most of those antibodies that got emergency use authorization are no longer allowed to be used because the viruses don't work. They don't neutralize the virus anymore. It's because it rapidly escapes from these.
Right, because the virus's whole job or its whole, what it wants to do is just keep replicating, right?
Exactly.
So it'll figure out how to do that.
And because it, as it's replicating its genome, it's putting errors in. Some of them are more fit, some are not. But if you also now make one that no longer binds to the virus, or let's say you have a large population, 90% of them get bound, but that one particle is able to get in, that's the only thing that's going to be produced at that point. And so that's the nice thing about non-neutralizing antibodies. It's probably more challenging to generate an escape variant because there's no selection at the upper end, right? You're not preventing the majority of the population from infecting a cell. All the viruses are still able to get in. It's on the back end. So it's not like you're going to be narrowing it down to a couple of particles that are now able to infect, and then all the progeny are now resistant to antibody neutralization. But I think people are starting to, back in the beginning when you were looking for monoclonal antibodies, the best way of screening is by looking for neutralization.
Yeah.
Right? Like it just makes it's an obvious assay to study. And in our recent work, we tried to evaluate other assays that could be used to do the screening that might identify non-neutralizing antibodies that are probably always there. We just never pulled them out because they were just negative in our neutralization assays. And so by doing some of these alternative assays, maybe we can identify these non-neutralizing protective responses that were underappreciated initially.
Are these antibodies that are the Fc antibodies usually lower affinity or not necessarily?
Not necessarily. There has been not a lot of work, but some work showing that lower avidity antibodies actually are better at inducing Fc effector functions. And that's because you might get more antibodies bound to the cell because you're not binding bivalently. So you're getting, instead of a really strong antibody binding to let's say the cell surface or the virion and using both arms of the antibody, that's only one antibody. But if you have kind of a lower avidity antibody, you're getting one arm binding. And so now there's space for another antibody to bind. And a lot of these effector functions require clustering of the Fc regions, the back end of the antibody. And so if you get more antibodies bound because there's space for these other antibodies to bind, you might actually be better at engaging these effector functions.
I'm learning something. But we should say that the Fc, that would come in with other cells attaching to antibodies that are on the target infected cell. Is that what we're talking about?
Yeah, so you would have to be, the antibody would be bound to its antigen, and whether that's on the virion itself or onto the surface of an infected cell.
Now, I think, are you talking about the back end of the antibody, the tail?
Yes.
Yeah. So the arms that Julie's talking about are the things that are binding the virus or the virus infected cell. And then there's, I guess, the tail or the bottom of the Y-shaped molecule. That would go onto the cell that has an Fc receptor.
Yeah, exactly. Sorry, I should have specified that. Yeah. Right. So that is also the angle of how the antibody's binding could dictate how accessible it is to an immune cell that's coming in, or like engaging complement. Obviously, there's a preferred like conformation, or a preferred like a hexamer would be better at inducing complement versus just some clustering. And it's that the tail, as you were calling it, or the back end, the Fc region, that's what engages to the Fc gamma receptors on immune cells. But most of the ones that are really effective at clearing the virus or clearing infected cells, these Fc gamma receptors are low affinity. So they don't just bind everything in circulation. They need it to be clustered to be able to bind very well. And that makes it specific. You don't end up just the cells targeting one antibody that just so happened to bind something. And so this way, you have the antigen, you have these antibodies that are binding, they're all clustered, and then that's engaging these Fc gamma receptors and leading to the activity that they can provide.
To whatever the immune cell is that's expressing the receptor.
And that activity probably leads to the death of the infected cell, right?
Yeah, so you can get death through like antibody-dependent cellular cytotoxicity. This is usually through NK cells. They'll degranulate, release perforin, and kill the cell. You also can get antibody-dependent cellular phagocytosis, which you have like a phagocytic cell, a macrophage, can come in and take up this infected cell and get rid of it. It almost like marks it for clearance.
And in some cases, it actually can be a bad thing because it can actually bring the virus into the cell. I don't think we're talking about that here, but it can happen.
No, I mean, it's something you have to always put out there. And people are, every time, like with SARS-CoV-2, and anytime you start putting antibodies into a system, it's always something you need to be aware of. So antibody-dependent enhancement. Mm-hmm. It's been well studied with dengue virus infections, where you have these cross-reactive antibodies, but they're not neutralizing, or you have sub-neutralizing levels of these antibodies. And so it's not enough to actually prevent the cell from being infected, but it now delivers the virus in an alternative way into the cell. Luckily, I've never observed ADE, as it's called, during alphavirus infection. So I don't think it's something we have to necessarily worry about, although it is something that always we try and keep in mind in case you see enhancement in some way of disease.
So are there any licensed vaccines against any of the alphaviruses?
Yeah, so there is a vaccine against chikungunya virus. The Bavarian Nordic, the VLP vaccine, it's called like Vinyana. It was actually generated or produced or developed, there we go, that's the word, by the NIH. And it works very well. You develop really high titers. It's a VLP vaccine, so it's not infectious. I think it's licensed down to like 12-year-olds 12 and older can use it. 12 or 16, I don't remember the exact age. There was a second vaccine that was approved, the Ixchix. That was a live attenuated vaccine. That was actually the first one to be approved, but it recently was revoked at the end of August because of some adverse side effects.
I can't imagine that's used very widely, right? Is it used for just people who live in certain areas or have certain jobs?
So I think it's mostly for travelers. So people who are going to be going into either endemic regions or regions with current outbreaks going on.
Okay, so military too, things like that, maybe?
Yeah, probably.
Yeah.
So one other thing I want to touch on briefly is that if you talk to folks about, let's say, influenza vaccines, one of the great hopes is that they can develop a universal vaccine, one that works on this year's virus next year's virus, the next year's virus, the virus 10 years from now, even though it's changed a lot of its surface. Are we at the point with any of the alphaviruses that's even something that's discussed, or do you think it's possible?
I do think it is possible. We've identified, the work that I've done, plus other work that is out in the world, these broadly reactive epitopes. So we know they exist. We know that it could be, it is doable. And some of the antibodies that we've studied, these monoclonals were generated following vaccination. And so it is possible to develop these broadly reactive antibodies. It's just a matter of trying to design the most appropriate antigen to elicit those broadly reactive antibodies. And the first step in that was to identify which epitopes we even needed to target before we can start designing a vaccine that might be broadly protective.
So Julie, our last question today is, so when you're not thinking about deadly viruses, what are you doing in your free time?
So in my free time, I spend most of my time with my 4-year-old son. Most of the time, I'm outside with him or we're playing in some degree. But I do like to bake if I have time and the energy to do it. That's something that I really enjoy doing as well.
So do you have a specialty, cakes, pies, anything that just sounds interesting?
So I am a big fan of the Great British Bake Off.
Okay. Oh, yes.
And so whenever that's on, it always inspires me to try really probably too challenging of recipes than I have the skills to do. I will say that I feel like I've done some really good French macarons at some point. And I tried making croissants. So I laminated some dough. So yeah, it's not the run-of-the-mill cakes. I try to be ambitious in my baking and go for fancy things that they're baking on GBBO.
I love that.
So how good is your chocolate ganache?
I think it's, it'd be good, but who knows what Paul Holly would say about it.
I love that. Our guest today is Dr. Julie Fox, Chief of the Emerging Virus Immunity Unit and alumni of the graduate program in the Department of Infectious Diseases. Julie, thanks for being with us.
It was great. Thanks for having me.
Links to additional information about Dr. Fox's research can be found on our Instagram account at PPPPodcastUGA.
As always, we appreciate you tuning in today. And if this is your first time listening to the People, Parasites, and Plagues podcast, we encourage you to check out the other exciting episodes launched last season. Please rate the podcast, and if you have any questions or suggestions for future episodes, reach out to us via email at ppp@uga.edu. This podcast is brought to you by the Faculty of Infectious Diseases and the Grady College of Journalism at the University of Georgia. It is supported by the University of Georgia through the Office of Research, the UGA Graduate School, and the College of Veterinary Medicine. Special thanks to our production assistant, Justin Nemitz, for research, editing, scheduling, and keeping us on track.